Thank you so much for coming out tonight. This is a fantastic crowd. Really happy for you all to be here. We're just going to give a brief introduction, and then we're going to turn it over to the panel. As many of you know, Vermont just signed into law the GMO labeling bill. Thought we'd get some applause about that. And so you might be asking, well, why are we talking about GMOs now? We've made this great progress, and yes, we have. And co-ops like City Market signed on wholeheartedly to have that bill passed, and we gave our support to it, as well as other great organizations like the Intervale Center. But really now is an opportunity and actually more of an imperative to talk about GMOs and talk about that technology and what it means to have it in our food systems. So that's what we're here to do tonight, to have a really non-political discussion about the science behind GMOs. So I hope you're all here for that. I want to make a quick save-the-date plug for a field trip that we're going to do related to the dish tonight. One of our panelists, Heather Darby, runs the UVM research farm up in Alberg, and she's offered to let us come and take a look at some of the cool things that they're doing at that research farm. It's going to be on Sunday, June 29th, and we're also going to make a stop at Blue Heron Farm, who produce some awesome pastured organic eggs for us. So sign up for that. You can RSVP on our website, and there's a little card on each of your chairs for that information. And before I hand it over to my colleague here, Joyce, I was just going to say, I have a beautiful baby daughter at home who's 8 weeks old, and it's been an awesome experience, and I didn't do that just for the ooze, but why I bring that up is because it's kind of cliche, but it's brought a new perspective to my life, just seeing her new perspective on the world. And with clear eyes, she's observing the world around her and seeing things for the very first time. And I bring that up because I hope all of us here tonight can kind of do that. You all might have opinions and beliefs about GMOs coming into this, and that's totally great. I don't want you to give up your convictions, but I want you to approach the panel tonight with some open eyes, just like my little girl. So, yes, thank you. Now I'm going to hand it over to my colleague, Joyce.
I just get to tell you about the fun stuff. So we have a Q&A portion tonight. So if you fill out a question card, which you'll find on your seat, we have ushers coming through, and we'll pass it up to Alison after our moderated discussion. And with every question you ask or comment you make, you get entered to win our fantastic door prizes. We have a wonderful City Market gift basket with local products. We have a $25 gift card.
Also a water bottle and a hat.
And a water bottle and a hat. And those will be announced at the end of the discussion, so that's an incentive to stick around. And then I also get to tell you about the wonderful ArtsRiot Solidarity drink that they're doing tonight. It's a Pim's Cup cocktail. So if you get one after the talk, $1 from each drink sold goes to the Intervale Center tonight. So and for the month of May, actually, that runs through Friday, May 30th. So thank you very much, ArtsRiot. And thank you guys. I'm going to turn it over to Alison Nihart, our wonderful moderator, and she will take it from here.
Great. Thanks, Joyce. Thank you, Todd. Thank you all for being here tonight on this lovely night. I know it's hard to come inside when we've had so few sunny evenings yet this spring. So as Joyce and Todd mentioned, the reason we're here tonight is to have a conversation about GMOs. And our hope is that we can dig into some of the science behind GMOs and hear from our experts on this. And we have some moderated questions we prepared, and we also have some question cards in the audience that we hope you'll write some questions down on and pass forward to us so that we can take those in the latter portion of the evening. And just want to sort of touch on what Todd was saying. We know this is a controversial topic. We don't think that's any reason to shy away from it. It's really important to have these public discourses. And we want to create a laid-back and respectful atmosphere tonight and dig in and answer your questions. So I'm going to start by introducing our panelists. We have Dave Rogers, who's a policy advisor at NOFA Vermont, Heather Darby, who is an assistant professor of agronomy at the University of Vermont, and Thomas Vogelmann, who's the dean of the College of Agriculture and Life Sciences at the University of Vermont. And before we jump in, I want to give each of the panelists an opportunity to introduce themselves by telling us a little bit about your professional and personal involvement in the food system. Just take a minute or two to tell us about that. And why don't we start with Dave?
Okay, well, thanks. It's good to see everyone, although it's a little difficult. So I'm, as Alison said, I'm a policy advisor with the Northeast Organic Farming Association of Vermont, NOFA. NOFA is one of the oldest organic farming associations in the country. And I like to say that we've been plowing the ground and planting the seeds of the local food system here in Vermont for a lot of years. My work involves lots of different issues at the national level, Farm Bill programs, National Organic Program, trying to work to get organic standards developed, local issues, state issues. NOFA was one of the organizations involved in moving the labeling bill. Before coming to NOFA, I was at UVM for 28 years in the College of Agriculture. And I was involved with various research projects and also taught courses in agricultural policy and issues, which at that time in 1985 were just starting to become something that agriculturalists were starting to think about. And so I've been paying attention to lots of things for lots of issues, agricultural issues and food system issues for a long time. Before that, I grew up on a farm. I come from a long line of vegetable and livestock farmers. My interests, I'm a gardener. I was a beekeeper for many years, and we'll be getting back to that very soon as I stop doing this. And like to be active and outside a lot. So I don't know if that's what you're
That's great.
for.
Thank you. Heather?
Okay. I'm Heather Darby. I work for the University of Vermont Extension and also for the College of Ag and Life Sciences. And I am an agronomist, which is essentially I work with crops and soils around the state of Vermont. I conduct applied research for the farming community, so essentially kind of trying to answer the questions being raised by farmers, and then I take that research and I deliver it to them in many different formats, from YouTube videos to research reports to field days, whatever we can do to basically get that information out to them. I work on a lot of different topics and with a lot of different farmers, from our traditional, conventional dairy farmers to our organic grain farmers. I have projects on hops, wheat, barley, corn, soybeans, and lots of other and quinoa now too. And then in my personal life, I also own a farm with my husband, Ron, in Alburgh, Vermont, a certified organic farm. We raise vegetables and fruit, and we also have some cattle as well. And that's about it. That sounds good. Huh?
Great, thank you. Tom?
Yeah. Well, greetings, everyone. Let's see. Rolodexing through, I guess, a description of what I am. I showed up in Vermont as a small tyke in 1955, moved out to Jericho in 1958, went through the Vermont school system, went to UVM, majored in biological sciences, and then with an academic track in mind, left the state for 30 years. And so initially went to Washington State University, got a master's degree in plant physiology there, then Syracuse University, PhD, again in plant biology. Then a year at in Wisconsin on a postdoc working on tree physiology. Then over 3 years in Lund, Sweden, where I worked on issues in plant growth and development and responses to light. First job was at the University of Wyoming in 1984. Rose through the ranks, got a phone call in 2001 saying there's a chairmanship position open, please apply. So I did. And then I had the opportunity to chair the Department of Plant Biology until 2008, in which someone said, why don't you try being in the dean's office for 1 year? I'm still there. So with respect to, I guess, the food systems, I would say our college is at the epicenter of food systems research in Vermont, and I'm just totally thrilled with that, and I'm thrilled about the future in terms of where we're going. Now, in my job, you work with a lot of agricultural leaders and practitioners of agriculture throughout the state. And it's a very interesting political terrain, to put it mildly. Some of the traditional large farms in the state readily have adopted new techniques in genetics, and they trust the science. But if you talk to them about the science of climate change, the conversation gets very agitated very quickly. At the same time, I'm looking out at this group, and I'm guessing that there probably aren't too many doubters about climate change, but much more hesitancy about the genetics behind what's going on in terms of agriculture and other areas. So what this means is, walk into a room full of people, I never know what to expect. With respect to other things, let's see, when I'm not working. Okay, full disclosure, people always ask me, do you have research in the college that's funded by large companies like Cargill and Monsanto? And the answer is no. We don't have research funded by a lot of other people either. So in full disclosure, if you want to make a contribution, we'd love to talk to you. When I'm not working, I'm usually in my vegetable garden. My goal is to get the size of the garden smaller than 2 tennis courts. I have about 30 varieties of garlic, and I'm having a tough decision in terms of which ones to keep and which ones to cut down on. I grow about 15 different varieties of potatoes, and so you can imagine the catastrophe that's waiting for me this summer, but I can hardly wait to get into it.
Thank you all so much for being here tonight and for participating on this panel. So we're going to start with a short presentation just to go over the basics of genetic engineering in agriculture. Full caveat, I am not a plant biologist, unlike Tom, and I am not an expert in this subject matter, but our goal here is to sort of bring everyone to the same page about some of the basics of the technology. So starting with some key terminology, really down to basics. Many of us are familiar with the basic concepts of genetic engineering. We could talk about just the science of it for weeks and years, but we're going to just cover the basics tonight. So DNA, we know DNA is building blocks of life. It's a molecule found in the nucleus of every cell that contains the recipe for making that organism. Genes are small segments of DNA. Each gene holds the instructions for how to produce a single protein. So why does this matter for tonight's discussion? Well, because DNA is a sort of universal language for life, that means that the genetic code from one organism can be transplanted into another organism, and that will express the same trait. So the originating organism can give a gene to another organism, and the recipient organism can express that same trait, which is what plant geneticists utilize as the technique for genetic engineering. So genetic engineering is the group of techniques that are used to cut up and rejoin back together the genetic material from one or more species to introduce the result into an organism in order to change one or more of its characteristics. And another term you'll hear is recombinant DNA. And this is DNA that has been genetically engineered to recombine genetic material from multiple sources. So that's the recombinant, is you're recombining it. A genetically modified organism, okay, we know that organisms are modified genetically by a variety of mechanisms, including natural mutations. But for the purposes of tonight's discussion, we're going to stick with the popular use of the term and mean organisms that have been modified through the use of genetic engineering. So GE technology has been used in a variety of sectors in addition to agriculture. And while these are pretty much outside the scope of tonight's discussion, there's enough covered just in food and agriculture. We wanted to just mention them to put GE technology in agriculture in the context of its wider use in other sectors. So in medicine, it's been used to make vaccines, insulin, human growth hormone. It's used in many drugs to create proteins and hormones used in medicines. And also, apparently, genetically engineered mice are used in medical trials. In forestry, GE technology is used to grow trees that are favorable for pulp, for paper and lumber production. In biofuels, it's used to maximize traits that facilitate the use of certain plants and algae also for biofuels. In food processing, it's used to create, for example, an artificial rennet that's used in the production of many cheeses. And of course, the topic we're here to talk about tonight, agriculture. And so for tonight's discussion, we're going to focus on GE in agriculture. So in agriculture, GE technology is also used in a variety of applications. Weed control, it's often used to introduce a gene that makes a crop resistant to a particular herbicide. For example, we hear about Roundup Ready. So Roundup is the herbicide glyphosate. The plant is not affected by that particular herbicide, so the farmer can spray that herbicide on the field and it kills all the other plants, but not the crop that you're trying to maximize the production of. Pest control, we're going to see an example of this on another slide shortly. A protein is introduced that is toxic to insects. Disease control, there's a few species of varieties of squash and papaya where a protein has been introduced so that it is resistant to certain viruses. And also, we have examples of adding specific nutritional components to a crop. For example, we hear about golden rice, which is the idea of adding an enzyme that's responsible for beta-carotene synthesis. We've heard certainly over many years about golden rice, but it's not currently being grown anywhere commercially in the world right now. And a more recent use of the technology is to support drought resistance in certain crops, and this is certainly something that will be talked about more as we hear more about the effects of climate change in agriculture. So I don't know how well you can see this from here, but it's basically a visualization of one example of GE technology in agriculture. And this is for using the BT gene. This is the protein that is toxic to insects. So they'll take the BT gene from, actually it's a commonly occurring soil bacteria, and they'll insert that into the crop, often corn or cotton, so that when a caterpillar eats the corn or cotton, it's affected by the protein and turns toxic in the caterpillar. So that's an example of how it's used in agriculture. Here's a few milestones in GE technology in agriculture. I'm not going to read through each of these now for the sake of time, but as you can see, in 1973, there was the first genetically engineered bacteria. And then there were some intellectual property rules in 1980 around patenting life forms, some field trials, and the first planting of the GE staple crops occurred in 1996, so it's been about 18 years since they've been grown in the US. So this is a graph of the top genetically engineered crops worldwide. I don't know how well you can see it, but I'll just narrate. The large blue section on the right is soybeans, that's 52%. Corn is the next largest section, 31%. And then a small amount of cotton and canola. And in terms of where genetically engineered crops are grown, the blue section, the largest again, is North America. So a lot of, the majority of them are here. South America is 35%, Asia is 9%, and a small amount of other. So as you can see, it's global, but North America certainly has the most. So just wanted to sort of set the stage with some of the broad areas of debate that are raised when people talk about GE crops. One is agrobiodiversity. This is the amount of different varieties of crops that we see grown in agriculture. Second is the use of pesticides and herbicides, and this is an overlap with the discussions about industrial scale agriculture versus organic agriculture, and genetically modified foods is kind of a subsection of that debate. Sometimes the issue of potential for cross-pollination with non-genetically engineered crops comes up. There's issues raised about the control of seed resources at a global level and intellectual property rights, and then also questions about the effects on consumer health. So we're going to start our conversation tonight with a few questions that Joyce and Todd and I put together ahead of time. And after that, we're going to spend time answering questions from the audience. So I see that some people are already handing in cards. That's great. Please write down your questions and hand them in, and we'll get to those in about a half an hour. All right, so first question. How does genetic engineering technology compare with other plant breeding techniques? So maybe you can help us put this in the context of plant breeding. And maybe we can start with Tom and move to Heather and then Dave.
Okay, sure. Well, putting things into context, this term genetically modified organism, it's the common lingua franca to mean something that's been made with recombinant DNA technology, but really crop plants have been genetically modified for hundreds, if not thousands of years. And so this is something we need to keep in the background of our minds. Lots of genetic manipulations, lots of collecting mistakes from the field. Those mistakes can be made through either cosmic rays or just mistakes in the DNA replication process. So we had a long evolution of crop plants. And what we're looking at today, in many cases, are plants that don't even resemble their ancestors in the wild. So we're dealing with highly changed organisms from what they were originally. I know of only 2 examples of where common garden plants, crop plants, have jumped the fence and gone back. Just about every other crop plant really requires us for the reproduction. So if we were snuffed out from the planet, they would go too. So that's just one thing to keep in the background. A variety of techniques have been used in breeding. For a long time, it was just sort of crossing different strains, different varieties, back crossing, very tedious kind of way to improve crops, sort of the Mendelian genetic sides of things. But after World War II, new approaches were developed. Once they figured out that DNA was the genetic code, they tried to figure out ways to accelerate the variation in the DNA and make more mistakes so that you could then look for plants with traits that might be beneficial for what you're trying to do. And so one of the most common ways that were used in the middle part of the last century was chemical mutagenesis. That's scientific jargon for saying that you soak seeds in an agent which causes a lot of mutations in the DNA. And of course, it's something that you probably don't want to do on yourself. But it's a random mutation kind of thing, and you have to sort of keep in mind that if you look at the number of genes in most creatures, the magic number seems to be about 25,000. So it takes about 25,000 genes to make a human, and it takes about 25,000 genes to make a flatworm. And one of the big revelations when they did the sequencing of the human genome project is they thought, well, we're such complicated creatures, we must have 200,000 genes. And as they worked their way through the chromosome and did the sequencing, the number kept getting smaller and smaller until a point where we were down to the level of flatworm. So where I'm going with this is if you use chemical mutagenesis, you're basically causing mutations in any number of those 25,000 genes, because most of those code for proteins. So I think you can get the idea that this is kind of like taking a shotgun blast to the genome. It's really hit or miss. You don't know what you're going to get, but it does increase the amount of variation that you get. And so you can, and I know that the wheat breeders out in Washington State, when I was a graduate student out there, they were constantly using this technique to come out with new varieties of wheat that would be resistant to rust. And they had about 7 years on average to make a new variety that was rust resistant. So it's this biological warfare that happens out there where the pests and the diseases are always out-tricking the plant defenses. And so this is also a challenge for the future. So flash forward to the last couple of decades when we got to really understand how to take out genes from creatures and move them around. This is the dawn of the age of genetic engineering, also biotech. And we've been at it for about 2 decades. And although we're focused in agriculture, their early applications were for medicine. And what I find fascinating about this discussion is that when people talk about GE technology and plants, they get kind of excited. It's like talking about politics or religion. You walk into a room, you start talking about your favorite GMO, and then people are all over you. But there's not that same kind of pushback in medicine. And so we have to really sort of step back and say, well, why are we treating these things differently? And so today, what we have the ability to do is identify specific traits. And we are commonly moving bacterial proteins into plants. And most of the, I would say, excitement about GE technology, and I'm using excitement in a global term, people are really excited and concerned, has to do with primarily 2 bacterial proteins that have been moved into plants. And this is against the background of tens of thousands of other proteins that are already there. So it's a fascinating thing. Now, what the GE technology allows you to do is, rather than have these very randomized techniques where you're depending upon chance to find a mutation that's beneficially useful to you, you can move gene of interest. And so you let your mind move forward as to what you might be able to do. And with any technology, it can be used for good things or things that you might have concerns about. So I'll stop there, because I know that you both have other things to say about this, and to round out the discussion.
Great, Heather, do you have anything? Sure. So
sorry.
the question was.
No, it's fine.
Tom answered it.
I know, he did. I was just trying to find something new to add.
So how does GE technology compare with other plant breeding techniques, and what's the context?
Yeah, well, I can, I've done a little bit of plant breeding on my own, and I've worked with a number of plant breeders, both that use, I guess what we would call very conventional methods of plant breeding versus sort of new techniques as well, and then sort of those new older techniques, the chemical mutagenesis that Tom was talking about. And I certainly have seen benefits to all and any one of those. I think one of the biggest differences that I see in just having a very small wheat breeding program of my own, I'm not a trained plant breeder, is time. And we've been working on a wheat breeding project. Really, the goal of that project is for us to develop wheat varieties that are more suitable to our climate here in the Northeast, because most of the varieties that our farmers here are able to access come from out west, come from places like Washington, where the climate is very different. And our farmers here try to grow those, and they don't perform very well at all. So we took on this project to essentially screen all of these varieties that we thought might do well here, and then pick out varieties that had traits that the farmers liked. So they were tall, they were weed competitive, they were good yielders, they were good quality. And then we tried our hands at actual wheat breeding, which is not an easy thing to do. And I'm sure at one point they moved wheat breeding out of the field into the greenhouse. Maybe that was controversial too, but we tried to do it in the field, where and this is a conventional process where we sat on 5-gallon buckets. I don't know if those are considered scientific tools or not, but we sat on these 5-gallon buckets, and with a pair of glasses on, we snipped open the wheat flowering part and essentially pulled out the boy parts, which I was very good at. It's called emasculation. Other people on our team were not good at it at all. I have no idea why.
Psychological emasculation is one thing,
I see.
but
anyway, and we pulled out the boy parts of the plant, and then what was left was the female. And then from that point, we essentially took some of the male plants that were flowering, and we dusted them on the female. And that was my first hand at plant breeding. Now we're 8 years later. The first try we had in the field, we had no successful crosses. So you can imagine, I'm sure this doesn't happen to real plant breeders, but you can just imagine we lost an entire season. Okay? And now we're 8 years later, and we've had 2 really bad growing seasons where we lost everything we had. And now 8 years later, we're still we're kind of almost there, but we still probably have a few more years of selection before we get something that's really stable. And so you can see this is like a 10-year process under conventional breeding, maybe even more, and that's if you find anything good, you know? And I can't even say we have anything that great, but it's ours, so we think it's good. But, and so with genetic engineering, it does really help speed up the process a lot. And that's really important, I think, especially in a changing climate, where things are changing really fast. From diseases to the kinds of diseases we have. I know I'm talking a lot, but just as an example, we have new corn diseases in the state that we have never ever seen before, and cornfields are dying from leaf blights. That there's no resistance in the corn varieties that many farmers are growing here, and so how do we get that resistance quickly so that we don't lose all our crops? So those are a couple of differences that I can think of just off the top of my head.
Great, thank you. Dave, do you have anything you want to add?
Yeah. So the genes are introduced into the cells through various methods, one of which is to load the gene into very microscopic pellets and shoot it into the cell, and those incorporate into the genome in a rather random fashion. And then there's a process of determining which cells have taken up the gene of interest. And then there's a long process of traditional breeding, bringing that gene into the varieties you're interested in. So the creation of the genetically engineered cell and initial plant and plants is just the first step. It takes years after that to do all the traditional breeding to come up with a variety that's of use and of value. So it does speed it up, but it's not a magic bullet. And then I would also say that there are other methods, you guys know about them, of course, other advanced breeding methods that do not involve genetic engineering that allow you to select for the traits of interest and to more quickly speed up the traditional breeding program. Marker-assisted selection is one of them. Tom mentioned that there are 25,000 genes, and I think we have over 100,000 proteins in the human body, which was a surprise at the time of the Human Genome Project, which means genes are helping one another out. There is, if you will, a sort of a microbial ecology of the gene. They help each other complete the tasks. And so when you have a process like this, when you have a process like this where the genes are being shot into the cells and into the genome, it could very well affect the expression not only of the gene of interest, but of other genes and how those genes are regulated and how they may assist one another. And that's what's been found, that there are a number of unintended consequences and collateral damage, if you will, that are really poorly understood. So I think that's a scientific challenge, big time, related to this technology and related to the other technologies of ionization and mutagenesis and all of that. So that's, I guess, what I would say.
Dave, that was a perfect segue into my next question, which is, and this one's going to start with Heather. So I wonder if you can each speak to some of the documented impacts on the positive side or the negative side of the various applications of GE technology in agriculture. And if you can speak to those various applications, that will help us understand which particular use of the technology we're talking about, instead of, thinking about it in a broad sense in all the various applications.
Yeah, I think I'll talk about this from a more applied aspect. The
Great.
Positives and the negatives from the farming community at a local level. I think. Well, I can say one of the negative aspects, maybe we shouldn't start with that, but one of the negative aspects that we see is plant and, weed disease and insect resistance to varieties or to the genetics that have been released because of poor management. It's not the plant's fault that it wasn't managed properly. And this is beyond just GMO crops. We see weed resistance to herbicides in conventional managed crops too. So it's really more about the management of those crops and less about the individual crops themselves, which then to me says that one of the negatives is also sort of a lack of understanding and education of the farming community at large that's using a technology that requires some specific management and very little, I don't want to say regulation, but very little oversight of if that management is actually being implemented. And so when you're growing it used to be actually, when you're growing a GMO crop, you're supposed to have a refuge of a certain percentage where that crop is not genetically modified, and it essentially kind of mixes up the genes so that you don't get resistance of an insect. And those refuges were not always planted. And so we started to see resistance. And then again, we also start to see resistance because if you continually use, let's say, a Roundup Ready crop, and you're continually spraying Roundup, then those weeds will mutate, just like we try to get our plants to do, and all of a sudden there'll be a weed that is no longer impacted by Roundup. And so that's just continuous use of the same herbicide and the same technology. And again, this happens regardless of whether farmers using a GMO crop or they're just keep spraying the same herbicide. So there's a big education piece, I think, and that's a downside of all of this. And some of the positives are that it has helped farmers control some really nasty pests that are out there, especially I would say in the insect world. And that's been very beneficial to many farms, I would say, some in Vermont and then beyond as well. So there are some positive aspects to that too.
Great. Dave, can you speak to some of the documented impacts?
So the issue of resistance to the herbicide, weed resistance to herbicide, is an important one. It's a growing one. There are now a couple dozen important weeds that are resistant to Roundup, and they populate or can be found on something like 60 million acres of American farmland, and that figure grows rapidly. So, of course, what you can do then is engineer the crop to be resistant to yet another herbicide. So if the first one doesn't work, hit them with the second. And those crops are, being developed and are ready to be rolled out. So I guess, a negative is, if you know about the history of this, and you alluded to it, American agriculture has been on a bit of a treadmill in many ways with respect to the use of technology, and this has not gotten us off the chemical treadmill. You can make the point that the chemicals we're using are less toxic, and that's true, but that's not to say that they are not toxic in various ways. So there is that. Those are documented. As an organic farming association, we're concerned about genetic contamination of non-genetically engineered crops, organic certified crops. That happens. It's happened with considerable frequency. It affects markets, particularly those who are growing non-GMO soybeans and corn for export to countries that do not allow any genetically engineered contamination. They test for any contamination, and there have been a lot of people who have lost entire crops that they were counting on to export because of such contamination. So that's an issue, and it's a real issue, and it's been a difficult one to deal with, both on the ground and also politically, and not only at a state level, but a national level as well. How do you address those issues? What are some practices and understandings that can be developed to stop that? So an economic impact. And, more could be said, but...
Great, thanks. Tom?
Well, so far I'm surprised that there's as much consensus on this panel around the issues as there has been, because this whole question about herbicide resistance is out there. It doesn't matter if you use GMO technology or conventional technology, weeds become resistant. And so looking at the agriculture and towards the future, the question is how we're going to beat the weeds at their own game, because if you're into large-scale agriculture, the two biggest problems are weeds and pests. And so with respect to resistance by the insect pests that are attacking things, in the late 50s, we were growing potatoes out in Jericho, Vermont, and I could run out with some rotenone and put it on the potato plants, and it killed the potato beetles dead. But they became resistant to rotenone, and then we ratcheted up a notch and used something else, and they became resistant to that too. And so get to the point where you start picking the potato beetles off my hand, and I don't wish that on anyone, because first of all, it's really a pain in the neck to do, and second, it's just not very effective. And then finally, we got these preparations of Bt toxin that you could spray on the plants. It's called potato beater. It worked okay. It's certainly better than picking the potato beetles off, but it wasn't that effective. And then now today we have a better formulation. It's called Spinocides, Spinocid, and it's a great stuff, but I'm wondering how long is it going to be, and it's organic, but how long is it going to be before the potato bugs become resistant to that too? And so this is a problem with any kinds of agriculture that we practice. It's how do you beat the pests at their own game? And so I kind of wonder about how could Vermont feed itself? And being a potato fan, I'd love to sort of imagine lots of potato fields out there. I'm just not sure how we're going to beat the potato bugs at their own game, because the only true and tried procedure that I've heard of that works is you let a bunch of guinea hens loose in your potato patch, and they eat all the bugs. But if we're going to grow potatoes on large scale to feed Vermont, that's a lot of guinea hens. And so looking forward, I guess, to the things that we are going to have to anticipate is that this is a game of biological warfare, back and forth, plant versus insect, plant versus plant. And then let's throw in some of the rusts and some of the fungal diseases, and how we're going to beat them at the old game, at their game. And so one thing that I think would be wise would be to keep every tool in the toolbox, because it's not going to be one thing. I think you made the statement that GMOs are not the single answer, not the silver bullet, or something to that effect. And
I think I said bullet somewhere.
Okay, silver bullet. Well, I happen to agree with that statement. It is one tool in the toolbox, but it can be used very effectively.
I wonder if I could just say something.
Certainly. Yeah.
I mean, this question of resistance, we've been talking about weed resistance and insect resistance. There's a selection pressure when you start to use this stuff.
Yep.
And in the case of BT crops, this insecticide is found in the plant, in every cell of the plant, so that when the insect bites a part of the plant, it will ingest some of this toxin and die. And that toxin is always there, and it's always being, the insects are always being exposed to it. Mm-hmm. And the resistance in that case can develop fairly rapidly and very broadly. In the case of BT used in organic agriculture, it's used very sparingly. It's used only when there's an outbreak and other methods haven't worked. So, I mean, this is one way of beating the insects at their game and keeping the tools in the toolbox at the same time. But anyway, I just wanted to mention that.
Yeah, I think that's it's really difficult, as you said, Dave, when you have an entire field that's essentially treated all the time to manage resistance, and that's why it's so critical that the management component is there, and I would say that's, in some cases, essentially what's led to the breakdown of these resistances, because the refuges are not being implemented to a point where they've actually created products now called RIB, refuge in a bag, which is the companies now mix corn seed from conventional varieties in with the GMO varieties, so that there are these plants that are interspersed in the field to some percentage level, and that was actually accepted, I believe, by the EPA as an acceptable refuge. And I will say that most entomologists, including myself, do not agree that that's an acceptable refuge. So there's some issues there. Big one.
So in the case of corn, a corn rootworm is developing resistance to BT corn. And so what is being done is several variants of BT toxin are being bred into the corn. So if the insects develop resistance to one form of BT, there's another form of BT already in the plant ready to kill them. But it's the same problem.
Yeah.
It's just a treadmill kind of thing.
Just one last note about rootworms. One of the best ways to control rootworm is to rotate out of corn. And that seems really simple, a simple practice for people to do. But there's lots of factors, around what these farmers are growing. And so, just as an example, I know in the Midwest, people thought, well, we'll rotate from corn to soybeans because soybeans aren't a host to rootworm. And then after I don't know how many years of doing this, now essentially the rootworms have adapted to making it through the soybean year, holding off for the corn. And so the strategy of rotating, even that has broke down because there's only a few crops now grown in rotations. And that's a sort of a bigger issue outside of GM crops and we talked about diversity. We don't have less diversity because of GMO crops. We have less diversity because of lots of other factors, but that does push for these crops to have more issues because they're grown more often, and breakdown of resistances to different diseases, etc. So, there's a lot of external factors happening here outside of GMOs. So.
Great.
We could go on this question.
We could go on for hours, I'm sure. Well, I'm going to pose one more question to you folks before we move on to the questions from the audience. And let's take about 5 minutes or so to answer this one. So maybe start with about 1 minute each, and then we can have 2 minutes or so to come back. So when a consumer sees a label on a product that indicates that it does not contain a GMO ingredient, or with the GMO labeling law set to go into effect in July 2016, if they see a product that says it does contain a GMO, what would you suggest a consumer thinks about in order to make a decision as to whether to purchase that product or not? What do you think should go through your mind in making that evaluation when you see a label that says it one way or the other? And do you want to start with that, Dave?
One of the great things that's happened in my working life is that food, how it's grown, what it represents, the values that are embedded in it are things that people think about and care about, and Vermont is first and foremost in that. So when we buy food, we're thinking not only does it taste good, but what's in it, how is it grown, how does it affect the environment, health, all that stuff. So I would hope consumers seeing a label would ask the question, well, what is this? And would, what does this signify, this label? What's this GMO stuff? Get the information, do a little research, get comfortable one way or the other with their decision. So, what I would hope is that they see that label and it triggers a complex sort of series of thoughts that they initiate.
Thanks. Tom, do you want to jump in on this one?
Yeah, okay. I think the challenge really is that the GE technology can be very widely applied. And so just having a label that says made with GE, I would ask the audience, what does it really tell you? Because it can be used for weed control, it can be used for herbicides, it can be used for as a pesticide, but it can also be used to add nutrition to grains and crops. Okay? Currently, there's research underway to add iron to rice because rice typically doesn't have enough iron in it. You can add other kinds of nutrition. For example, in order to get all the amino acids that you need to be a healthy person, you need a whole slug of them. But if you're a vegetarian, it's very hard to get tryptophan and methionine. And so you eat beans for those things, but you better make sure you have a lot of beans. And so thinking about what could be added to other grains to be making them more nutritious, you could add proteins with more of those essential amino acids. You can take the allergens out of plants. You can reduce the level of natural toxins that are present in plants, because one misperception that we have, or one thing that we don't really worry about, is that we tend to assume that the plants that we eat in a store are just completely all safe. But a curious thing is a lot of them come from some fairly toxic plant families. One thing that I see quite often when I walk through health food stores, and I do this sort of as a hobby whenever I travel anywhere, I go through a health food store and I sort of check them out. But one thing I commonly see is heirloom potatoes, which are the kinds that I grow. If they're not kept in pitch blackness and they're exposed to small amounts of light, they turn green. It's the chlorophyll that makes them turn green, but they're also making solanin, and that's a nerve toxin. And if you eat enough green potatoes, they're bitter, so it's not like it sneaks out of the woods and gets you. Very, very bitter. You're getting quite a load of something that's probably not good for you. A lot of people don't recognize the fact that mangoes, you can have an organically produced mango. It's a member of the poison ivy family. And so if you're very, very sensitive to poison ivy, you better be careful about eating mangoes and also pistachio nuts and cashews. And so thinking broadly about the technology, are we worried about the issues as to how the technology is going to be used, or are we worried about the technology itself?
Thank you. Heather?
I would agree with that.
Yeah, one of the questions that I hope we could ask the audience actually is, why are you here? Why are you maybe for or against GE crops? Is it because you think that they might hurt you, or is it environmental issues, or is it you don't like Monsanto, or is it, what is it? I mean, just curious, because I think that's what you would likely think about, like Dave said, when you go to purchase something that might be labeled. Like, what is it about GE crops that just doesn't settle well with you? And I think that's something to really think about. Is it, and that goes back to, how your food is produced, and if you really want to know everything about how your food is produced, then you likely are not going to be buying that crop, that material anyway, because it likely, maybe wasn't produced locally per se, or it might make you question the practices somebody's using. But if you just say maybe, well, I don't like the way that this impacts farmers in other countries, and so I'm just not going to buy it. So I guess to me, it's what, how do you feel about these? What is it about these crops that, just it doesn't settle well with you? And I'm assuming most people are here because maybe it doesn't settle well with you. And I asked myself that same question. Well, how would I feel when I go to the grocery store and I see that? And I think it really does come down to, what is it about that you it's making you feel like you care?
Yeah.
Right?
I think we'd be having a much different conversation, and people's views of genetically engineered crops and foods would be very different if we didn't start out with engineering herbicide resistance, engineering toxins into them. If we started out ramping up vitamin levels, eliminating toxins and all of that, and there was a label on there, people would preferentially choose it perhaps. When the Flavor Saver tomato came out in the mid-90s, they couldn't keep it on the shelf. It had some attributes that people were looking for. In Hawaii, when they have genetically engineered papaya for sale and it's local, people will buy that preferentially over New Zealand papaya. So it's a complex process of thinking about all these attributes. And I just think that the companies have shot themselves in the foot in their interest in selling their chemicals, that they were always the chemical companies. If they'd started out doing some other things and showing some real benefits to consumers of these crops, there'd be a different calculus in the mind of people who are buying.
So I'm going to go down a little bit of a path here based on Heather's request and say let's hold off for about a minute on the questions from the audience. These are good questions, so I do want to get to them. And what was your question, Heather? What brought you here tonight? And maybe it can just be popcorn style from the audience, so not even full sentences, but just what is the one thing that you're interested in learning more about, and that way when we're answering these particular questions, you can answer them with a mind to the issues that people have come here with tonight. So just shout it out.
Toxin residues in food.
Great. What else?
Impact on biodiversity.
Impact on biodiversity. Great.
How do we know what's bad for us?
How do we know it's bad for us? And the first one was toxins in food. Yep.
Projections for future labeling.
Sorry, it was what?
Projections for future labeling.
Projections for future labeling. Go ahead.
Knowledge of what's in the food, GMOs, not GMOs, for my clients as a chef.
Great. From a chef's perspective, knowledge about what is in food, GMOs or not GMOs.
Simplicity.
Simplicity.
And being able to get our heads around, as on a human scale, the processes in our food. It's become so big.
Understanding the processes in our food, given the scale of how food is produced, it sounds like it's what you're saying. Great. Anything else from the audience you want to share? These panelists can keep in mind as they're answering the questions. Go ahead.
How do we know they're safe when they're so innovative when we don't have any history?
How do we know they're safe when we don't have as much history and they're on the cutting edge, perhaps, in terms of science? Go ahead.
If something is labeled GE, is there a way to know what that means?
Is there a way to know what it means that something's labeled GE? Corporate control in the food system. Anything else? Yeah.
Just for producers of produce or other crops and being able to save the seeds, and the legality of trying to save seeds that are GMO, or having the problem of cross-pollination of GMO products with heirloom varieties or varieties that are not GMO.
Great. So from a farming perspective, the legality of seed saving and also potential issues around cross-contamination with heirloom varieties and non-GMO seeds. Yeah.
It's such a complex area. This is one little window that gives us a chance to educate the public and to educate ourselves.
Complex, yes. And I will mention now that Joyce and Todd are going to be posting some resources on the DISH website as a way to dig into more information. If this is only whetting your palate tonight, you can go online and do we have a sense of when those will be up, Todd?
Get them up by the end of the week.
By the end of the week?
And also, willing to post all the questions that we're not able to get to tonight, and maybe have the panelists chime in with some written answers.
Great. All right, well, speaking of audience questions, let's jump into the first one here. So this question is about allergies to GMOs. Considering that GMOs are not tested in standard dermatology patch tests, how do we know anything about whether these foods might be causing allergies in people when they eat them? Does anyone have a thought on that?
I do, but go ahead.
Well,
Go ahead.
they you know, the companies do testing on the GMO crops before they're released. And one of the things that they do test for are common allergens. And there has been cases, for example, there was a case, I believe, with DuPont and Pioneer where I believe they were trying to insert a Brazil nut gene into soybeans or something, and they were able to catch that allergen because they do test on common allergens. I think one of the areas where people are really concerned, scientists included, is that these unintended consequences, sort of Dave was talking about, allergens that aren't common allergens of people, those aren't tested for. You know, there's not that kind of rigorous testing. There is some testing done, I can't remember the term for it, but where they actually take the materials and digest them in certain digestive fluids to see if they break down and things like that, just to see if there might be something there. But the GM crops are tested for common allergens. And I guess, I don't know if there's been a lot of documented cases of acute allergens or acute response to GM crops. So, you know, I think it's been 20 years almost, and there hasn't been any acute toxicities, not that there isn't going to be any unintended consequences that we know of. And also, as this continues to happen, and there's these things that Dave was talking about, you know, things are so rapidly changing that it doesn't mean that there isn't a chance for that to happen. So, I don't know if that helps to answer questions. So it's not an easy answer because this technology is changing so fast. You know, the genes are being mutated, et cetera, and new things are coming out on the market. And so the chance is still, you know, there. We don't have testing for that. So,
Dave, you want to follow
From the very beginning, the concern about the production of novel allergens was expressed even by FDA scientists and others as well. Even the National Research Council and others have said, look, you know, we may very well be sort of activating some pathways in plant cells that would yield some heretofore uncharacterized antigens. They're not common antigens. We don't even know what they are. How could we test for them? And because we don't, we really don't know what we've eaten, and because, I mean, the only way you're going to tell if something is really toxic is if you have a situation where someone has an acute illness following shortly after ingestion, so that you can connect the dots very clearly. The physician can, and others. But if it's a very sort of subacute or subtle effect, and you don't know what you're eating to begin with, because you don't know whether the corn proteins are genetically engineered or not. Very hard to make the case. Very hard to connect the dots. Very hard to do the epidemiology, which is one of the arguments that people make in favor of labeling. People will know that they've consumed, let's say, a genetically engineered substance. It's something that they will perhaps share with their physician or whomever. Maybe you can start to develop some information about it, but there have been a number of cases of such novel antigens having been developed as a result of the genetic alteration that is occurring. I mean, there's a lot of research that needs to be done.
Tom, do you have anything you want to add to that?
Yeah, first of all, the observation, can we guarantee that something is 100% safe? And the answer is no. In this issue of allergens, for much of the genetic engineering that we're talking about, you're talking one or two proteins. An enzyme that gets around the metabolic block imposed by glyphosate, and that's taken out of a bacterium, or the BT protein. And so you can test those things. And we need to put this in the context of the background of all the other proteins that are there in a plant, okay? So going back to this idea of 25,000 genes, 10 to 15,000 different proteins are present in a plant at any given time, and start adding them up, okay? So you have some spinach, that's 15,000 proteins of some kind. Add lettuce, well, it's another 15,000. Some are similar, some are different. Let's have a piece of chicken along with that, another 15, 20,000. So, you know, just playing the game of odds to sort of worry about whether one or two proteins that now have been added are going to be a problem, the odds are pretty small. Now, it doesn't mean that we shouldn't be diligent, but I'll add also another layer on this, and that is that in our digestive tract, we are hosting more bacterial cells than there are cells in your human body. And you should be very thankful that they are there, because they control our immune response, they control our digestive processes. Those guys are living and dying all the time. Hundreds, thousands of different species. They die, another 10,000 proteins that go off into your system. So we're talking huge numbers of proteins that you encounter every single day. So is it 100% safe? No, but it's not 100% safe on conventional breeding either, because things can happen there as well. And the irony of all this is that there's more testing of GE crops than there is for conventional. You don't have to test conventionally produced new varieties. So 100% safe? No. What are the odds? Not very good.
I would say that the international bodies, Codex Alimentarius and World Health Organization, have put forth protocols, suggested protocols for screening for allergens, and they're not really used in screening our foods. We can do a much better job of determining the allergenicity of constituents in our foods, both GE and non-GE. So again, a lot of research needs to be done.
We both agree that putting the protein from a Brazil nut in a soybean probably isn't a very good idea.
No, because it's a well-known allergen. But you know, there's some very interesting results that have occurred in recent years where something that was not an allergen and introduced into this was in the case from a bean plant to a pea. A related plant, this protein became an allergen when it was introduced into the pea. So genes are not just created when they're transcribed, but they're sort of in each plant they have this sort of processing that goes on. And apparently, what happened in this case is, even though the gene was the same and the protein looked the same, it was processed in a different way by the cell, and it created an allergen effect. So, I mean, you know, a result like that is something where you say, geez, you know, we have a lot of work to do.
Well, and I think that's part of the, you know, some issue that people raise is that they're commonly not actually testing the actual plant for, you know, screening the plant for the allergens itself. So, you know, that's an issue.
Yeah.
Or can be an issue, especially based on what you just said.
Mm-hmm. Interesting. All right, so this question says, is there data showing yield increases and or reduced agrochemical use on GMO crops? I hear these claims, as well as the food security feed the world argument for GMOs, which seem dubious. And I'll invite whoever wants to jump in first to do so.
Go ahead. No, okay. Yeah. Well, I mean, there's been the holy grail is to increase yields, but all the easy work was done back in the era of Norm Borlaug and the Green Revolution. And so, if one looks at the ability to increase yields, I think we really have pretty much approached a ceiling of sorts. There's only so much productivity that you can squeeze out of a crop plant. And having said that, there's a lot of energy that's going into engineering rice, and they're trying to make the photosynthetic pathway of rice more like corn. And so they're talking about moving dozens of genes over, and they're hoping to greatly increase yield there. So I'm not sure that this whole argument is about increasing yield, because the easy work has already been done, and I think it's going to be very hard to go further than that. But what we are looking at is moving into traits that will give more environmental resistance to plants. The reason why that the corn crop wasn't a total disaster a couple of years ago when the drought hit the Midwest is because the genetics have gotten so good at allowing plants to survive on less water. So we're going to be seeing a lot of that. With respect to herbicide use, I've seen figures that globally the use has gone down, but we have to, again, look at a case-by-case scenario. If you are using correct management practices, you can reduce the amount of herbicide that is used. If you use incorrect practices, then you get different kinds of insects moving in and different kinds of other things. And so I've seen data going both ways. So I don't know, Heather, do you want to jump in?
Sure. I work with a lot of colleagues across the Midwest and the East Coast. I work with corn specifically, and we all concur that we don't see yield advantages to growing GM corn. And in my own test trials at UVM, I've never documented a consistent yield increase from growing GM corn either. And, that's just one site, so it's not to say that in some year, and that's many years of data actually, not just a single year. I've also never seen a quality increase from that corn either, so increase in the quality of the feed. And others, like I said, other big corn states, corn agronomists, other places have said the same thing. So why do we continue to use them? And I do, it's a very complex question. I don't know if you want to get into all of that, but so we haven't seen a lot of documented year after year increases in yield and quality. And in terms of herbicide usage, I do think that initially that was one of the promotional pieces of GMOs, especially with Roundup, is that if you grow a Roundup Ready crop, you can use Roundup, which is very inexpensive and also a very effective herbicide, but also a very short-lived herbicide in the soil. And in the past, a lot of farmers in Vermont were using an herbicide called atrazine, which you may have heard of, which is a very harsh herbicide that has a lot of regulations nowadays. It's not as used as much, but it's still used. What I found just through my work with farmers is that farmers are not just using Roundup, and that they actually still use 2 herbicides to grow those crops, and sometimes they'll grow Roundup Ready crops and not even use Roundup. So, and this is again a bigger, more complex issue of why they're growing that corn to begin with, and some of it has to do with risk and insurance, and we could get into that more. But I would say there's been a reduction in herbicide use.
One interesting idea is, your slide indicated that there's been a tremendous consolidation in the seed industry in the last 20 years since this technology became available, and global chemical companies bought seed companies so that they could have this suite of seeds and chemicals that would work together. And in developing the genetically engineered varieties, they have also brought in the advanced genetics from traditional varieties. So the point I'm making is we've had some years of intensive breeding of genetically engineered, let's say, corn, to put the best genetics into those genetically engineered varieties. Any increases in yield are due to genes and breeding that are unrelated to the genetic engineering traits themselves, and yet the increases in production often are ascribed to the fact that they're genetically engineered. So you have to sort of tease that apart, and I think that's an interesting, I know we're not going to talk about politics, but it's an interesting, it's just an interesting dynamic that has occurred in the industry.
Well, I mean, but it's not politics. It's real.
Yeah.
The fact
Everything's
that DuPont owns Pioneer, which is one of the largest seed companies in this country, corn seed companies. Syngenta and Monsanto, DeKalb and Monsanto, these are all corn companies, and Mycogen and Syngenta. So when I used to do a comparison trial of GM versus non-GMO corn yields, and I would ask the seed companies to submit traditional varieties, and they would initially. And then I think the third year into it, they started giving me just Roundup Ready corn instead of the triple stack corn, because that's what they were calling conventional. It was only had 1 gene in it versus 3. And now if I call one of these companies, and not every company is like this, but some of the big companies, and you ask for a conventional hybrid, the choice, and this is real, I called this year, there were 2 that fit into our climate for Mycogen and 2 that fit into our climate for Pioneer. And if a farmer, and one of them I would never grow. I mean, I've trialed it before and I think it's a lousy variety. So, you know, when you have hundreds versus just 4 to choose from, it makes it very difficult for a farmer to choose from 4, and when 2 aren't really even the best genetically, whether they were GMO or not. So there's a little bit of a lack of choice out there for the farmers, and oftentimes, you know, they're picking varieties that they know performed well before they were had added traits to them.
And they've discontinued traditional lines oftentimes.
Yes, they have discontinued.
Heather, before we go on, can I just ask you to define triple stack for the audience, the people who might not be familiar with that term?
Yeah, so a triple stacked corn likely has 3 types of genes in it, or more. Sometimes there's quad stacks bred into it. So it might be Liberty Link, which is a type of herbicide resistance, Roundup Ready, and then it might have some of the insect resistance as well.
Last year, Monsanto came out with a sweet corn, and they basically first marketed it heavily in the Northeast, and it has a gene for glyphosate resistance and 2 Bt toxins. So you know, it's probably in your store, but you don't know that.
One last thing, the corn companies are hearing the demand change. I will say that from the Midwest to the Northeast, you know, consumers hearing it is really important. If it's really something that people don't want in their food, then stating that and making that choice does change what farmers do, what stores do, and also changes what companies do. And so if you think that if you feel strongly about something, you can make a difference. And just in the last few months, certainly those companies they're sold out of those varieties. There's an increasing demand for non-GMO corn seed, and as that demand increases, it's not like Pioneer got rid of all their conventional lines. They just haven't had them on the market, and that is rapidly changing. So you should feel some success if that was maybe your thing.
But at the same time, we don't want to make this too simple. Beware of unintended consequences, because if you go the GMO route or the conventional route, that means you might be using more sprays, more herbicides, more pesticides.
Yeah, I wanted to make the point about one of the consequences of GE is it has allowed the monoculture model of production to continue. I mean, if we had not been able to do this, we would have been using some very nasty chemicals to a point where the environment and the public would not stand for it, and some serious change would have happened somehow or other. So we're sort of in a period where we're getting away with using perhaps less toxic chemicals for a period, but GE has continued to allow this, I think, unsustainable monocultural model of agriculture, where we could, if we just learned a lot more and put a lot more research into plant-pest relationships, understanding soils, plant-soil relationships, we would discover things that would enable us to sort of have productive agriculture in ways that were not so problematic.
Well,
yeah. Well, I was just going to say on both sides that I do think it's really important that we all have a really good understanding of what each one of these crops does and the benefits to that. I will say that it has become increasingly important to the farming community, not only in Vermont, but elsewhere. And the reason I know this is not just because I work with farmers here, but other places as well, And also, all you have to do, now that you folks get these magazines, is pick up a trade magazine. A trade magazine either put out by John Deere, the Furrow, or International, or one of the farming magazines, and they are loaded with articles on building healthy soils, which just astounds me. And so there is, this is working its way into the farming community, and what farmers are finding is if they really take care of their soil by growing cover crops, by doing a multitude of practices, that some of these other tools aren't really needed, including G crops. And I invited a farmer to a conference we had this winter, and he's milks 900 cows. He's a very large farm in Pennsylvania, and he's very into building soil health and also using no-till. And many of our farmers here think that they have to use GMO crops to be able to farm no-till. And so somebody in the audience asked him, what GMO crops do you use? And he said, I don't use any. Because he personally felt that it went against what he was trying to do in terms of building the health of the soil. And so this sort of different mindset of actually taking care of a larger ecosystem is really infiltrating the farming community from all kinds of avenues, including seed companies that are making a killing off of selling all this really high-priced cover crop seed. Mm-hmm. So there's a lot of really great things happening. People aren't just growing GMO crops.
Yeah, I'm really glad you brought this up, Heather, because I don't think that anybody seriously thinks that we can go on our current ways of doing things and have a sustainable agricultural future. At the same time, we do have this problem of feeding ever so much more people on the planet. I think the solution would be a lot easier in terms of sustainability if there weren't so many of us. But having said that, the question is what technologies are we going to use? Because when we think about sustainability in the broadest sense, you're absolutely right about the health of soils. I mean, this conversation's been going on since the 1970s, and we're finally getting around to doing something about it. But I think it's, again, keep all the tools in the toolbox, because it might be something in the middle.
We have a very short amount of time left, and I'm going to ask this one question, which I think could be as simple as a yes or no answer, could warrant a few more sentences. Will the noise around GMO and the labeling debate impact the, albeit limited, funding of ag research at UVM? And maybe this is something Heather and Tom can... Limit it? Is that what you said? Well, Tom was alluding that we are potentially limited in actually the total amount of funding that we get at UVM for ag research. And so the question is, even though it's limited, is there a chance that the GMO debate, the fact that, perhaps I'm inferring now, but the fact that Vermont is in the spotlight for GMO labeling, could this affect the resources that are coming to UVM for ag research?
Oh, I'd love to see the country get worked up to the point where they double the size of the USDA research budget.
And change some of the priorities as well.
Well, okay, we can agree about that. But the issue that we're facing right now is that when I look forward over the next 20 to 30 years, we're seeing some of the most, I would say, challenging set of environmental issues on this planet. And so being a person that sort of is grounded in science and trying to think about giving choices for the future, a lot of research needs to be done. Of course, you're all expecting me to make this pitch because I'm the Dean of the Ag College, but I'm also doing it because I'm a scientist as well. And I think there's lots of opportunities for research to really help build a more sustainable future. I don't know whether the labeling law is going to cause that kind of willingness to invest or not. We'll have to wait and see. It's probably not going to come at the level of the state, because this is a very poor state and they're doing the best they can.
I would say something. For the last two farm bills, we have worked with other organizations around the country to increase the funding for classical breeding, plant and animal breeding, in the USDA. Perhaps I'm overstating it by saying that biotechnology and related sciences, very important work, very interesting work, but it has somewhat sucked all the air out of available funds that could be made available to explore some of these relationships that I mentioned earlier. And we have been unsuccessful in getting the kinds of increases that really would help organic farming for sure, to be able to address some of the problems that exist, and some of these larger problems as well. So maybe it would affect research availability at UVM by providing some support for classical breeding and some of these other areas of research that are unrelated to advanced biotechnology. And you guys could, attack those issues of sustainability the way you're saying needs to happen.
Great. Well, we are just about out of time. This has been an absolutely fascinating conversation tonight. I wonder if you could each end with, if there's one thing you hope the audience will take away from tonight's discussion in a sentence or two. What is that? I know it's a lot to summarize.
Sometimes I say I feel fortunate to have arrived at this age I am at, and to be so optimistic.
You're younger than I am.
Well, so you're less optimistic. But I mean, in the years I was at UVM started in 1974, and seeing the changes that took place over the 28 years I was there, I saw incredible evolution of interest in food, interest in agriculture, young people being engaged in it. We see this at NOFA all the time. It's incredible. The only way I can keep my optimism up though, is by I can't leave Vermont, So, no, just kidding. But I feel optimistic. We're talking about things openly. We understand problems, and there are a lot of people who are engaged with this. Certainly, with the GMO issue, people came out of the woodwork to support that. That would not have happened 20 years ago. And I just hope that kind of engagement with food and agricultural issues continues in our society. Sorry, that was more than one word.
You're speechless? Yeah, well, again, I guess what I hope the audience walks away from tonight with a better appreciation over the complexity of what we're dealing with. And so what I would hope is that we sort of focus on the issues moving forward. What is it we really want to accomplish when we talk about sustainability? What are these tools that we're going to need to meet the challenges with pests and diseases? And one thing is certain, we know that they're going to be there. And so rather than focus on the technology, I think it would be more productive to focus on the issues.
Yeah, I agree. I feel like I hope you learned something, but I feel like it probably drew up more questions than answers, which this debate often does, and it is really hard, I feel, to find the information that most people are looking for to help you feel one way or the other, to help sort of answer all these questions you have. It's really difficult, I think, to get that information because a lot of it is, from one side or the other, and it's very sort of heated, and there's always references and things, and so it makes you feel sometimes like it might be the best source and maybe not. So, you have our contact information, and certainly, I would personally invite you to ask myself questions if you have other questions. But I agree with Tom and Dave. I feel very optimistic about agriculture in Vermont and the changes that I'm seeing in farming practices. And one of the things I think that I've tried to do is really focus on best agricultural practices that help the farmers remain viable, but also are protecting the environment and all of our natural resources. And I think finding those key practices is really important, and it goes well beyond this discussion that we're having today. So.
Great, thank you all. And thanks to everyone who was here tonight and contributed questions. Sorry we didn't get to them all, but as Joyce and Todd said, we might be able to put some of them online, and you'll also be able to find potentially some of the resources that our panelists have been able to utilize if you want to further your education. I know ArtsRiot is serving dinner tonight. They have Malaysian food, and want to thank them for being our host. And I think I'm going to turn the mic back over to Joyce. Oh, yes, please. Thank you.
I would just say thank you all.
Thank you.
And thanks to our fantastic panelists. You guys did a really awesome job. That was really fun. Thank you.